Well, thank you very much for your kind introduction, and thank you for inviting me to present to you about some of our research and sort of an overview over the field of plasticity in the auditory cortex. So when you're sitting here and listening to me right now, your brain is working hard because it has to adjust quickly to certain things. First, my voice is different from Christine's. Then I speak English and do it with a Californian German accent, so you have to get used to all these type of things. And there are additional aspects like the room acoustics and whatever. So there are lots of things that your brain is trying to extract from the acoustical information that goes into your ear. And we have heard about the very difficult and elaborate mechanism that happen in the inner ear to translate that acoustical information to some neural activity. But then you have a whole brain behind it that's trying to make sense of that and translate that acoustical information into meaning. So what does it mean, what is being said to you, and what it may give you as insight. So, and that's what we will talk about a little bit today. It's not this quick adaptation. So you do this adaptation within a few minutes, you sort of get used to the speaker and say, okay, I got it. But that it's based, the ability that you can do that is really based on a lifelong learning experience that you had in other environments with other speakers. And so there's a long-term, slow learning process that enables you to do this successfully and hopefully with an auditory system that is fully capable. And we know if you have a hearing loss, if you have other issues that relate to changes in the brain, those effects will impede your ability to extract that information. So we will talk today a little bit about these long-term adaptation effects or learning or plasticity that result in changes in our brain due to the environment, due to your learning, due to the mechanisms that are being invoked. So what this sort of supposed to tell you here is that it is not it's your brain that hears, not just your ears. Of course, the ears are necessary, but there's a lot behind it. And a fundamental task of the mammalian brain is to deal with these changing conditions in our sensory environment, short-term, long-term, and we can do this by the processes of adaptation, sort of a quickly adjustment, assessment, and based on these very long learning effects and that are much more slowly developing. So these processes result in plastic changes that change actually the structure and function of our auditory system over short time and long terms. And so I'll give you a big overview of what we will talk about a whole bunch of things, actually. So first, we will give a slight overview over several systematic representations of auditory information in the auditory cortex, as how it relates to frequency, stimulus frequency, and the timing. Then we will move on. That these representations will undergo changes depending on the environment. As we mentioned already, learning is important for new sounds, but there are also changes in the structure of our brain with aging, with all other kinds of influences that also result in plastic changes in our brain that compensate maybe for those effects in order to optimize our ability to hear. Next, we will talk about, oops, that's not working. Okay. How these adjustments are governed by different plasticity mechanisms. There's not just one type of plasticity, and actually there's several types, and we will go over those in some detail and talk about how they change your ability to hear throughout your whole life. So that's what we'll do in these, about these plasticity processes and how there are expressions and what their potential underlying mechanisms might be. And the goal of this is to illustrate that some of the challenges and opportunities that we have, that the role of plasticity plays in our ability to shaping and maintaining good hearing ability, good hearing health, so to speak, throughout our lives. Okay, so let's start out with a few general properties. We heard from Professor Petit. Most of the information was really down here in the cochlea, the transduction mechanism, and with all its complicated processes that go on. But from there, we move through the brainstem, the midbrain, to the auditory thalamus, and end up here in the auditory cortex. And that's really where the place we are talking about today. And the main reason is that many of these plastic changes that we will talk about are mostly or most strongly expressed in the auditory cortex. It doesn't mean there are no plastic changes here in the auditory system below cortex. For once, the cortex also projects down. This big arrow here means everything from the auditory cortex is being rerouted to lower stations. So any plasticity effects up here will affect things that go on subcortically. But the strongest expression of plasticity and the main mechanism for plasticity actually are located in the cortex, and so that's why we will focus on that station. So we'll give you a little bit overview over the sort of a few fundamental aspects that you should keep in mind while we're going through this. And the first one is a very simple principle. Each neuron in the auditory system and auditory cortex is really sort of a frequency filter. It prefers a narrow range of frequencies from all the frequencies that come in from the outside. So here's an example. So what we're plotting here is from a single neuron somewhere in the cortex. We play different tone frequencies. Each of the little lines here corresponds to the response of a stimulus that either has this frequency or this loudness, this intensity. So for example, this one here is a 4 kilohertz tone at 5 dB SPL, very soft tone, and you don't see anything except this little line. But sometimes you see these little blips here. These are the action potentials, they are spikes that have been generated by that stimulus. You can see most of those combinations don't do anything. There's a few occasional spikes, those are called spontaneous, they happen anyway. But you can see here's an area where we see pretty much every time where we play this 8 kilohertz and that intensity or that intensity, we see a spike. And if you sort of look at that with a little outline, it's sort of a V-shaped type of area. That means this neuron here in particular likes, we pick this frequency down at the tip as its characteristic frequency, which is something like 8 kilohertz, although the frequency range is a little wider here at higher intensities. So it likes not just 8 kilohertz, but I don't know, 10 to 3 kilohertz or something like this. So the preference depends a bit on the intensity of the stimulus, but roughly we can characterize it as saying, okay, well, this neuron likes something around 8 kilohertz. And this is a similar representation, just the sum of many of these frequency response areas or tuning curves. And you can see that it sort of has a V-shaped type of thing. Again, we can determine the best frequency here or the characteristic frequency, but we can also measure how wide that range is over what frequency range these neurons respond to. Now, in cortex, of course, there are lots of different neurons that respond to different frequencies. And if you put it all together, you find that there are these maps that occur that you can find in auditory cortex. So this is the cat auditory brain of a cat. All this colored gray area here, light or dark gray, is dedicated to hearing. So the brain of the cat is particularly broadly engaged in processing hearing. What we are looking at is what's called A1, or the primary auditory cortex, which is really the station that is the first input from subcortical station. From there it goes to other regions in the auditory cortex for further processing. But we don't concern ourselves too much with the other areas. We just think about this for now in this primary auditory area. Now, if you look at this area here, you can find that each location and each of these polygons here corresponds to one location that we recorded from. And you can see that there's a color gradient, and the color gradient corresponds to the frequency that location prefers. So you can see these magenta, purplish frequencies are on this side, high frequencies. These yellow, orange things, low frequencies are on this side. So you see this very nice, what they call a tonotopic gradient or tonotopic map, cochleotopic, relating to the frequency orientation in the cochlea So it's a very nicely organized map along, at least along this direction from low to high. Along this direction, this vertical, you can hear these are all blue. They're all like something like whatever, 10, 12 kilohertz. It seems to be they're all like the same thing. So why do we have this dimension? Is there something else represented in that axis? And indeed, that is the case. If you look along this map here, these are a couple examples of tuning curves. And the CF, the best frequency, we plotted here, the characteristic frequency. And now we're not plotting what frequency it likes, but what is the range of frequencies that this neuron, each neuron location at these locations corresponds to or likes. So there are neurons that are on this scale here that show something about the width here. The blue one means it's very broad. That means the neuron responds to a wide range of frequencies at high intensities, whereas the red area here, the red range corresponds to neurons that really are very specific about the frequency range that they respond to. And when you look at this, it looks a bit confusing, but if you follow this line here, again, with these dots here as guide a bit, so these are all the blue neurons here, meaning 10 kilohertz, but you can see some regions here where you see the orange color, meaning they are very specific to that 12 kilohertz, whereas the blue areas here and here and here, there the neurons respond to a much broader area. So this dimension here from, so the cortex is really divided in two dimensions. One is the frequency axis. The other one has something to do with the preference of the frequency range that is being processed. These two parameters we'll come back to later, so that's why we talk about them a little more here. Now, cortical organization in general, of course, is complicated. The neocortex is a layered structure that is about, from the cortical surface down to about, depends on the species, and humans maybe about 2 millimeters thick, in rats maybe 800, 900, 1,000 micrometers, or about 1 millimeter thick. But along this axis, you can distinguish very clear certain structures, and people have assigned layer numbers to it. So layer 1 up here, 2, 3, down to layer 6. And these different layers can actually be distinguished by the content of what cell types are in them, where they project to, where they receive projections from. So those are very specific organizational principles, which we'll ignore mostly. But so most of the data that I will show you, the cortex are from layer 4 or 3, and that's the input layer from the subcortical station. So it comes up, ends up in layer 3 and 4, and then it goes on to processing in the upper layers. The upper layers project to other cortical regions, or it goes down to the lower layers, which then provide feedback to the subcortical stations. So not only are there different cell types and so on, but also across cortex, there are different, in principle, there are two different types of neurons. So the excitatory neurons. That means they put out glutamate as an excitatory transmitter to the next neuron, whatever they're connected to. And those are mostly the pyramidal cells, these little triangular things. These are the cell bodies of those excitatory neurons. And then they're also here, indicated in this column, are the inhibitory neurons. These are local neurons, mostly local. They project to, mostly at least, to these excitatory neurons, but they use a different neurotransmitter, as GABA, which is providing inhibition of the activity of the following neuron. Inhibition means the membrane potential of these neurons will be go into hyperpolarization. That means they cannot produce a spike as easily, an action potential, whereas glutamate provides excitatory input that will depolarize the cell, and then it can hopefully produce some kind of action potential. Of course, these neurons all are sort of form circuits, local circuits within the auditory cortex. Here's just an example of sort of a mammalian canonical circuit in the primary auditory cortex. It's A1 again. And you can see here the layer numbers, 1, 3, 4, 5, and so on. And MGB, this is a thalamic output arrives here in layer 4, goes into some kind of neuron, like a pyramidal cell, which then projects up to layer 3, that goes to layer 2 goes down to layer 4. So there's an intrinsic mechanism or intrinsic circuit that calculates or produces transformation of the information depending on what these circuits are supposed to do. Of course, we don't know exactly what they're supposed to do. We know they're there. What they exactly are doing is not so clear, but we know they deal with maybe sharpening any of these aspects that I mentioned before. Maybe they be tuned to aspects that relate to the voicing or whatever of what you're listening to. Nonetheless, this circuit exists, and of course there are also some of the inhibitory neurons involved. We see that in a minute. These are just a couple examples of, there's a pyramidal cell excitatory neuron here in layer 5 or 6 with lots of dendrites going up to the upper layers, and then eventually an axon going out to the next station. So let's look a bit at the very local circuitry, and that's the one I already mentioned. We have pyramidal cells. These are the excitatory output cells. They receive input from a thalamic cell, so from many thalamic cells actually that go here, have an excitatory input. But thalamic input also goes to some of these interneurons. In this case, it's called the PV interneuron. There are different types of interneurons. We won't get into that. But this is provalbumin positive. There's a certain protein on the outside that identifies the cell. But nonetheless, this is a neuron. When it's active, its output will suppress the activity of what's going on in the pyramidal cell, will hyperpolarize it. So there's sort of the fight between how much excitation does it get from the thalamic neuron and maybe other cortical neurons, and how much inhibition does it get from local interneuron. And those two inputs fight with each other, who is going to win out, and that determines whether the neuron is going to fire or not. So we can measure these things by intracellular recordings. You can determine what are the synaptic currents that each of these inputs will induce. So you can do that by clamping these cells and seeing what is the current when there's an inhibitory input, what is the current. I think there's an excitatory input. So here you see a depolarization, there you see hyperpolarization, depending on how much these synapses are being providing. So these pyramidal cells then receive excitatory and inhibitory inputs, and combined, these synaptic events determine whether this cell, this pyramidal cell, will actually fire an action potential. Here's an example of these currents. This is a membrane potential of a pyramidal cell. And so here we have an inhibitory postsynaptic current induced by some stimulus here. And at the same time, we also have an excitatory current. And these two will combine to give you some type of membrane potential. And in this case, excitatory input is almost as big, but the inhibitory input is much reduced. So maybe you get a spike here or not. That depends on the threshold of this neuron. But it's this combination of these two inputs that is relevant. Now, this distribution of excitatory and inhibitory inputs, of course, depends on the frequency selectivity of the neuron. So for some inputs, we look at this here. This is now, we have different stimuli, different frequencies. And this is the input to pyramidal cells. So, and we can look at the excitatory inputs. So this is our pyramidal cell that gets input from the thalamus, but it also gets input from this inhibitory cell that's sitting next to it. And you can see in this case, both neurons, both inputs, the excitatory input of the pyramidal cell or the inhibitory input are code-tuned, or they match in their tuning. So when excitation is high, green here, inhibition is high too, and they follow each other very nicely. So if you look at a stimulus here at the CF, characteristic frequency, that's what you get. You get excitatory input, you get inhibitory input, but together they create an action potential. But if you take a frequency that's a little off here, where the drive is less, so especially for the excitatory input, you may not get an action potential anymore. Here's another potential match of these two things, or mismatch. In this case, excitation is nicely tuned, narrowly tuned, but the inhibition is much more broadly tuned, so there's a bit of a mismatch. The reality is somewhere in between these two things. So some neurons are more like this, some neurons are more like that. But it's a combination of these two inputs that really determines what's going on. And that's really all we need to know at this stage. So let's move really to the questions of what type of plasticity are there. So the first one is the most commonly known one. It's called associative plasticity, and it is induced really by active learning. Everybody here has undergone this plasticity. Whenever you learn something acoustically, and of course it goes for every other sense as well, let's say you travel to another country and you have to learn something about that language and see now what are the Italians saying. So you will learn to discriminate and identify those sounds. They're different from ours, or from the one you're used to, and so you have to put some effort in to making better auditory discriminations between those sounds. And that is, as you know, takes effort, takes learning. You have to sit down and you need to be rewarded for it. If you don't get your reward for it, you won't learn. So most of these learning processes, associative plasticity, are based on, okay, here's a task. If you get it right, you know, you get a reward. Now for us, it's not usually a sugar pill or so like it is for our animals that we train, but it's an internal reward. You know, you feel better about yourself and you're motivated to learn, and that puts out some, or activates some kind of mechanism that sets this thing in motion. Now, what is that mechanism? But before we get there, let's just quickly sort of summarize the points here. So this is the ability to reorganize cortical circuits through experience. So that's the main point. And as an adult person or animal, that's learning. And it's possible throughout life. You learn from preschool till we die. And it will change the numbers and strength of synapses and the connectivity between the neurons that we have talked about that will be influenced by this plasticity. So it depends on what is it that you learn. So examples are learning to play a musical instrument, of course, learning to speak a new language. There are lots of auditory things that you learn. For example, you move to your new house along the Mediterranean and there's this water rushing to the beach all the time. You will learn that this is a background noise and you will adapt to that, and you know, okay, I don't pay attention that much anymore because you can sleep again after a week or so. So it happens in many different circumstances for audition. It's inducible by operant conditioning, and that's, again, you get a reward. Now, the reward doesn't always have to be positive. Many of the animals, you get actually a little negative reward. It's still a reward, but it's has a negative implication. Nonetheless, learning is affected by it in slightly different ways, but it's still learning. And it has to do with the release of neurotransmitters like acetylcholine, noradrenaline, or dopamine, serotonin, many others, that regulate this reorganization. And we will talk about this in a minute. And it's also really the inhibitory processes play a big role in how that is being adjusted. And we come to this in a minute. And again, it's active throughout life. Okay, so let's look at an example here. So this is a study by Dan Pauly and others. So what they looked at a frequency map in the rat. And we remember here, this is the gradient in color, corresponds to the preferred frequencies. Red in this case was high, blue, low frequencies. That's a normal animal. And let's just look at the upper part. This is primary auditory cortex. This is a different field. It doesn't matter that much. So, and then the animal, or another animal, was trained to pay attention to 4 kilohertz tone. So whenever it heard various tones, whenever a 4 kilohertz tone was played, the animal had to push a button, get a reward, and it does it for about a couple of weeks, and the animal gets good at it. So after about 2 or 3, 4 weeks, it gets it every time, or whatever, 80, 90% of the time gets it right. So now you look at the auditory cortex of an animal that has been trained, and what do you see? Well, you still see a frequency map from low to high, but suddenly this 4 kilohertz here in gray, you see many more locations that correspond to that frequency. So somehow the cortex has expanded that frequency that was associated with the reward. So this is one of the consequences of plasticity, an obvious consequence. You see a map change. Now, there are many other changes that are not so easily displayed that relate to plasticity, but this is a simple essay to find out, okay, what is changing and how can we measure that, how much is changing, and things like this. So this is a reward only given when correctly recognizing that stimulus. So you have to do it right. If you do it randomly, this will not happen. If the reward and saying correctly, was 4 kilohertz, you won't see that change. There are more complications with that on the long run. Maybe someone will ask me about it later, but that's not so relevant at this point. So we talked about mostly frequency organization, but there's another aspect of hearing that is just as important, and that has something to do with the timing of the events that arrive at the central stations. So when I speak to you, there's a certain rhythm here in my cadence, and it's about how many words do I speak per second, or how many syllables per second. And it's something in the range of 4 to 10 events, so to speak, individual events per second. So it depends on how quickly someone is speaking. You have to get used to that, of course. If someone speaks fast or slow, you got to adjust to that as well. And indeed, you can show that cortex will reorganize itself depending on what type of timing is imposed on the stimulus. So here's an example. So here we plot this is a time of the beginning of the stimulus. And the stimulus consists of these little red lines. Each of them is a little noise burst. And you can see here at this is the speed or the frequency of these noise bursts. Up here, they're on 20 hertz. You can see they're following each other quickly. Down here, 2 hertz, there's only one here, one there, or 4 hertz here and here. So they're far spaced. Here, they're very closely spaced. And you can see these black, it's a little hard to see, but these gray black spots here are actual potentials that were created by that stimulus. And you can see here around 10 hertz, you see every time there's a red line, you see a nice response. Also at here, lower repetition rates. But if you go to, 15 hertz, it's only every second. Here at 20 hertz, it's not that much anymore, that the following ability is reduced. So the neuron is still capable of doing this 10 hertz very well, but not so much or less at higher frequencies. So then this animal was rewarded to learn to pay attention to, let's say, 20 hertz repetition rates. And so whenever it got that right, it got a reward. And now you can see here in this experimental animal, 20 hertz is still, you get every time you get a response, unlike here where it's more sparse in between. So you can also increase your ability to pay attention to these temporal patterns by learning. And that's just here summarized. This is the repetition rate. This is the number of spikes that you got. And in the control, this is the open circle, that's what you had. So maximum was around 10 hertz or so. And for the trained animal, it moves a bit higher. So you do a better job at 15 or even at 20. It's not too overwhelming, but we have other experiments I haven't shown here where we trained, this is in rats, where we train monkeys in doing the same task. And they can go from, again, in a normal monkey, you see 10 hertz or so. In a trained animal, suddenly, you see neurons at 5 to 50 hertz to 100 hertz, just as well as here for the 10 hertz. So you can change the temporal properties in the cortical neurons quite dramatically using these operant conditioning learning aspects. So how does it work, I think? I mentioned already that there are many different neuromodulatory systems in the brain that influence the degree of plasticity or whether plasticity is actually taking place. So I mentioned there's the cholinergic system, there's a nucleus spatialis minor somewhere, that if that is activated, it will release acetylcholine throughout the brain, throughout the cortex. And that release is associated with operant conditioning learning. So when you learn one of these systems, it could be the cholinergic system, also the serotonic, noradrenergic, dopaminergic system, they all slightly different sources, different type of neurotransmitter, and they do slightly different things. They're not doing exactly the same type of plasticity, but they're all involved in some type of plasticity. So when you have a probe that measures the amount of acetylcholine free-floating in the cortex, you will see when the animal's learning, it goes up. So now one can make use of this type of mechanism that is certainly implemented. If you want to study how plasticity works, let's take one of these systems. Well, before we get there, just how that works sort of on a little circuit base. So if you have a target stimulus here that says, oh, the animal's learned, okay, this is important, I better get it right to get my reward, it will, this nucleus basalis, for example, will release some neurotransmitter in the cortex, acetylcholine, that will go on one of these inhibitory interneurons. And then this is one of these, there are several types. This is a somatostatin inhibitory interneuron, this is a parvalbumin inhibitory interneuron, and they inhibit this pyramidal cell. But this acetylcholine will activate this other inhibitory interneuron that actually, if this being active, it will increase activity it will decrease its activity here, because it's an inhibitory interneuron. It will decrease activity of these other inhibitory interneurons. And as a function, this pyramidal cell will be able to fire more. So you suppress its inhibition by this other inhibitory circuit. So it's complicated. But that's the way these neurotransmitters usually work. There are some kind of intermediary effects on the inhibitory system. This is just a reminder, we talked about this before. We have these tuning curves, and we can get these tuning curves for, again, frequency and tone intensity. You can measure the excitatory synaptic conductances that comes from the synapses that are going to a pyramidal cell, but you also have the inhibitory synaptic conductances. In this case, you can see that the excitatory and inhibitory tuning curves look very similar. So you can see that these two excitatory inhibitory inputs were very well matched. The example we showed earlier, excitation and inhibition are nicely matched for the whole tuning curve. This is just a different plot of the same thing. This was done by one of my students, Andrew Tan. And then you can see this nice correlation of the excitatory strength and the inhibitory strength. If excitation is high, inhibition is high as well. So you see this nice covariance between these two inputs. Okay, so now what happens when we do, we release acetylcholine in the brain artificially. That means we stimulate that nucleus that releases acetylcholine. That is for us much more convenient than having an animal going through training and while it's training, record while it's doing. It's a long process. The training is slow. The learning is slow. What we can do with this sort of artificially, we can just stimulate that nucleus that releases acetylcholine, and we can immediately look what's happening. So it's a very sort of time-compressed way to see how the mechanisms might work. So what do we see? So here's an animal. We stimulate the nucleus basalis, we release acetylcholine to the cortex. Then we record from auditory cortex. Now, what we do in the cortex, we record excitatory and inhibitory currents. We do this with wholesale patch clamping that allows you to discriminate between these two things. And so first we look at the response of two tones. Before we do this, then we pair a tone, specific tone, with this release of neurotransmitter, step number 2. And in step number 3, we measure again our tuning curve, now without the pairing. So after this pairing is about for 5 minutes, 3 to 5 minutes only, that the learning is taking place. And then we see, well, what has been changed, what has been altered. These are experiments that were done with Rob Fromke in the lab. And so here, that's what we see for the excitatory currents. These are different frequencies. This is current, and we can see 2 plots here. One is pairing. Here, this is number 1. And we can see this neuron something like 4 kilohertz. So then, in this step number 2, we paired a 2 kilohertz tone with this release of acetylcholine. And when you measure then after here in step number 3, you measure now what is the tuning curve, excitatory tuning curve of this neuron. You can see it's now shifted to 2 kilohertz. So after only 3 to 5 minutes, the excitatory things already shifted over. So the preference of the neuron shifted from 4 kilohertz down to 2 kilohertz. So here's the same thing when it comes to inhibition. So before step number 1, we look at inhibitory currents. It's the dotted line here. Well, it prefers 4 kilohertz. So that means excitatory was tuned to 4 kilohertz, inhibitory was to 4 kilohertz. That's what we would expect. So now we pair this 4 or 5 minutes. And what we can see is actually here we see at 2 kilohertz for the excitation an increase in the currents. But for the inhibition, we see a decrease. So that means during this learning process, inhibition is lowered, and that's just the time course of this. But so excitation and inhibition change in opposite directions. Now, what does that mean when it comes? I mean, this is sort of instantaneous, but this is sort of the second part of the story. These things change over time. So initially, here, this is our cell before we do anything. We look at excitatory and inhibitory currents. So that both are tuned, in this case, for an 8 kilohertz frequency. So excitation, black dots, is tuned. Inhibition is tuned. So they match. Now you pair these frequencies, and you look at excitation and inhibition. And again, even right after or within the first 30 minutes after pairing, the excitation's already moved to the new location, which is, I'm not sure, 4 kilohertz. But the inhibition is still at the old 8 kilohertz, the strongest, but it's reduced here at the new location. So this is after 30 minutes. Now you look at 100 minutes. Excitation is still at the new one, 4 kilohertz, but the inhibition has started to move over. It was best here at 8 kilohertz. Now it's best at 4 kilohertz, or it's somewhere in between. And then if you look about 3 hours later, now the excitation and inhibition are rematched. So what you have to do, what we have seen is that excitation and inhibition are sort of disengaged from each other before they're matched. Then you see a mismatch between the two. And then after a while, the inhibition moves back to the new frequency that is now preferred by this neuron. And so it creates, again, a status that is sort of stable, the excitation and inhibition unmatched. So within 3 hours, we have moved, this neuron has learned to prefer this new frequency. And we see that by having matched excitation and inhibition before, the mismatch in between, and after 3 hours, they're matched again at the new location. So that's basically the mechanism that is active in this associative learning type of processes. You have this manipulation of inhibition through the neuromodulatory system, either acetylcholine or noradrenaline, that do similar things, not exactly the same, but they're very similar. Okay, so that's sort of the mechanism. Now, does it really work? I mean, we know, of course, when we train an animal for 3 months, we know that animal now likes this new, whatever we trained them to like, 4 kilohertz tone or whatever, was rewarded for it, and we see this over-representation. So one criticism of these experiments was, well, you do it for 3 minutes, 5 minutes, they don't learn anything. It's just the local effect that you can observe, but it really doesn't have any meaning when it comes to the perception of the animal, by the animal. But it turns out, actually, it is meaningful to the animal, even after these 3 to 5 minutes pairing, that the animal is doing better at whatever task we asked it to do. Here's an example for this. So these were rats that were trained, listen to sounds, and tell us whether the sound is there. Simple experiment. And in this case, what we changed was intensity of the tone. And if you look at the black, that's what you normally see. These are the correct responses by the animal after. So we trained these animals before in this task. So they do very well. He had loud tones. A little less, he had medium loud. And not so well, he had 30 dB loudness. So it gets only, whatever, 25% correct responses. So now we paired a 30 dB tone for 5 minutes with this release of acetylcholine. And then we measured again. The animal was awake, then was going back into its apparatus and telling us what it heard. And then now what you can see, the red curves, that's what we see after this 3 to 5 minute training. From 25% before goes up to 70% correct. So it clearly now knows, yeah, this soft tone, that was important, and it hears it. So it is really, even this very short process does change the perceptual ability of the animal. So that's just to show that this actually works. Here's an example of the temporal plasticity, where we trained, well, a little more complicated. So we tried to figure out, and this has to do with cochlear implant studies that we did. So we have, the animals are deaf, and the only stimulus that gets in goes through electrical stimulation of the cochlea. So you put an electrode in, you stimulate that, and different rates of repetition of those pulses that go in. And in the normal animal, so if this is the repetition rate, and this is cumulative probability of getting responding to that repetition rate. The black curve here is sort of in the middle. Let's look at the 50%. It likes it. 50% of the population is about here at 8 hertz repetition rate, and half of them are faster and half of them are slower. So after the animal has been deaf for a while, not using its auditory system, it deteriorates. And the result is that if you look at the blue curve, this curve shifts to the left, and meaning now 50% is only here at whatever, 6 hertz. So 50% of these neurons are less fast. They don't like 6 kilohertz. But the shift to the left means the ability of the neurons to follow has slowed down. And then we trained these animals with a regular learning task to detect these faster repetition rates, and rewarded when they were detecting them. And if you look at the red curve, now it shifts not only to the right, it actually shifts a bit beyond the normal repetition rate. So the animals are now, with electrical stimulation, better capable, have a higher capability of following these repetitions for this electrical stimulation. So these were experiments done by Marcia Reggio, and Professor Reggio is actually sitting there, and Ralph Bartel. And this is a cochlear implant type of application of operant conditioning. Okay, let's move on to a different type of plasticity, and this is maturational. So maturational is, everybody has heard about this probably, but we have sensitive or critical periods early in life that allows our brain to adjust to what it's supposed to do. So we know that from humans, language learning, for example, is important. It's easy for you to pick up a language as a child. If you're exposed to whatever new language, you'll learn it, you pick it up in no time. I mean, lots of kids go to bilingual kindergartens, they pick up all these different languages, no problem. If we do this, good luck. We have to work on this. But I mean, sensory critical periods are present in lots of things. In visual system, if you block one eye, you are almost blind after that. So if you don't get that input, that stimulating input, the statistics that are relevant during that critical period, your sensory performance as an adult is diminished. So here examples are, for example, in cochlear implants in deaf-born children. If you implant them with a cochlear implant early on, when they're 1 or 2 years old, they'll learn normal language. Implants are not the most high-fidelity devices. Nonetheless, they can learn a language just fine. If you do that on a kid that is 14, 16, 18 years old, it's not going to work. The critical period is over. And vision deprivation can lead to blindness. So what about this critical period? Now, there's no learning involved. It's not like these animals that undergo during the critical period. They're not being rewarded for hearing it right. There's no feedback. They just get the statistics, and it's really in the statistics that will reshape the sensory mechanisms, central circuits. And if the statistic isn't right, you will have a deficit. So this is active during the critical period, which depends on the animal, of course. Postnatal days 1 to 20 in a rat. For us, here is an example in humans. It's much longer, and there are different stages when language learning is happening. I can't even read the other thing. What did I say? High cognitive functions, sensory, yeah, well. Anyway, so there are these moments in life in which that mechanism is active. And of course, it shapes the functional organization by aligning excitatory inhibitory input again. So we're coming back to this alignment that we talked about before. And here's an example how that works. These are studies in Merzenig's lab with various Sidani, where they looked at these are the cortical maps that are frequency maps that we already talked about at different age stages. So here is at 11 days after birth in a rat, you can see certain frequencies, whatever, 10 kilohertz are there, but not much. At 12 already, you get an expansion, you see a wider range of frequencies, and here at 14 days old, you see the full normal map. So this is sort of the maturational development of these cortical maps. And of course, you can disrupt that. If they don't hear normal environments that have normal ranges of frequencies and whatever in their environment, this will not happen. So for example, if you put a kid, a baby, in a neonatal, what are called, these boxes where they are kept under good conditions, often they have very noisy fans and noise backgrounds. And that really has been shown if you present rats with these noises or constant noise, they will not develop these maps. And once, of course, they're adult, that's it. I mean, whilst you can work on it to restore some of that hearing, but it's very difficult. So you want to do this, you want to keep the environment that you, the babies, grow up in have the right statistics. So avoid noises or give them rich environments that they can soak up and make their connections develop to deal with those ranges of sounds. So I mentioned already that this also has something to do with excitatory inhibitory balance. And here's an example of an adult animal. So we reiterate, so these are the excitatory currents, frequencies, strength of current, inhibitory. They're not the same values, but they're tuned about to the same frequency, whatever, 4, 8, 6 kilohertz. But if you plot how they relate to each other, whenever excitation is high, inhibition is high too. This is excitation versus inhibition. You see a nice correlation in the strengths of 0.87 or something like this. Well, here's an animal, P14, young animal in a critical period. You can see excitation is nicely tuned, inhibition is not. So here you see this correlation between excitatory currents and inhibitory currents there's no correlation. They're independent of each other. So in adult, they're nicely correlated. In a young animal, they're not. And this is what happens to this correlation over time after birth. So here we had 14. The correlation is very close to 0. This is a correlation varying 0 to 1. So in animals around age 15, rats again, the correlation is very low. And here in this time here, third week roughly after birth, that's where the correlation is really being established. And then it reaches adult levels after that time. So it's this rematching or this matching up of excitation and inhibitory tuning that sort of closes the critical period. And it's really that match because you can accelerate that. What you can do is you can present tones over and over again during that time. And you can close this period earlier. And that is, when does it close? It closes when the two inputs, the excitatory input and inhibitory input, are matched. So this matching, once they're matched, plasticity is over. This type of plasticity, the maturation plasticity, is over. So this is during the third week. This work again by Rob Fromke and Anja Dorn in our lab. So that's sort of, again, coming back to this balance type of thing. Here's a bit more complicated issue, but it's really related to the same thing. We're talking again now about temporal plasticity. So the timing of the responses in cortical neurons also is changing during the maturational period, critical period. So here's an adult animal. Same experiment we saw before. We present clicks or noise bursts at different different rates, 2 hertz to 12 hertz or something like this. And this is a response to the 7 hertz repetition rate. You see every time where there's a little red dot, that's a click, you can see a nice spike. This is several repetitions. You see a nice response. And this is a pyramidal cell. This is an inhibitory cell at that age, an adult animal. So you can see actually that follows very well as well. So in adult animals, both of those follow very nicely these repetition rates. If you look at the young animal here, so these are red dots are the stimuli. This is a pyramidal cell, and it gives you a response to the first click, but not to any of the others. So these pyramidal cells don't like these quick, fast repetition rates in the young animal. If you look at this one here, this is now what is fast spiking neuron, inhibitory interneuron. The inhibitory interneurons follow very nicely. So it's a big difference between what excitatory neurons can do and inhibitory neurons can do in the maturing rat. In the adult rats, they're matched. So there's some mismatch here, and one can actually show what is the cause for that the pyramidal cells cannot follow. And the reason is really, here we just saw that the inhibitory neurons follow very nicely. What really the problem is that the inhibitory responses in the young animal, these are the red lines here, they're very long time courses, whereas in the adult animals they're very short. So it's the duration of inhibition that is maturing very quickly from a very short duration in the adult to still very long duration in the young animal. Because inhibition is so long, it just cannot, the next stimuluses are still swamped with inhibition and it cannot follow. So it's this maturation of this duration of inhibition that increases the ability of the neuron once it reaches adult age. And it actually can be shown this is a GABA B dependent mechanism, but I don't want to go into that. Nonetheless, it's again, we're talking about this balance between excitation and inhibition that is also behind the temporal plasticity during maturation. So I want to give you an example of something relevant to us, actually, how maturation plays a role in our ability to hear. So these are behavioral consequences of maturational plasticity. And the point here is that we can improve our signal detection in noise if we are exposed to the proper stimuli during maturation. So what am I talking about? Here, this is in a rat, of course, this is not in humans. So what we have here are spectrograms of rat vocalizations. There's different rat vocalizations we just put together and played them to the animal. And what you get are these are 100 repetitions of these stimuli. And you can see at certain times, you get every time when this thing starts a sound, you get a nice response. You see these nicely lined up straight lines, meaning those are strong responses to these vocalizations. So that's what you see in most neurons with the right frequency. Okay, this is what we see when we add some background noise to these vocalizations. So the vocalizations are still there, but now we have some kind of noise that goes on and off. And if you do this, and this noise is changing from one trial to the other, so you can still see some of these vocalizations respond, but much less. So there are only a few that survive this noise effect. It's a signal-to-noise ratio of 0 dB. That means the vocalizations and the noise have about the same intensity. And 0 dB is for us, yeah, it's okay. We can just still work on it. Minus 10 dB, when the noise is 10 dB louder, we really get into trouble. Okay, so what we did though is we exposed some rats to a specific type of background noise that had this structure, temporal structure. And we exposed them at a moderate level during the maturational period. So just played that on 24 hours, wasn't very loud, so they could hear other things in the cage. Certainly didn't damage anything. And then we measured this again. Okay, so here's our, just the response to the noise. This is vocalization response by itself. This is modulation. This is modulated noise plus a vocalization, just for an animal that was exposed to this noise. And you can still see, again, their responses to vocalizations here. But it turns out that in the naive rats, you get about, what is here, this is population, the percentage of neurons that where you still see some residual responses to vocalization. It's about 10% in normal, whereas in those animals that were exposed to this specific noise, it doubles. We have many more neurons that are still capable of hearing, seeing that response in the background noise. So something has changed. Somehow the system adapted to the structure of the noise. And now when you play a vocalization, it looks like the animal should be doing better. Well, is it doing better? So we had to do behavioral experiments and see, is this animal actually better in detecting signals in that specific noise? And it turns out it is the case. So let's look at, these are just, we trained them on detecting either this vocalization or that. Let's look at this one here. So the naive animal that hasn't been exposed to the noise does pretty well here at a very low noise level. You know, it gets 85% or so. And then if you go to 0 dB signal-noise ratio, it drops down to 75, 72%, and then it deteriorates very rapidly. At -20, nobody can really detect those things anymore. So these are the animals now that were raised in the noise. So yeah, it's still in a normal case. Here at 0 dB, they still do just as good, just as well as they were without noise. And then eventually it drops off. You know, nobody can do better than that. So, but clearly, just by exposing the animals to the statistics of the noise, they weren't rewarded. There was nothing going on. This maturational period allowed the animal to learn something about the statistics of the noise, and then adjusted the system so that when another signal occurs in that noise, it could still pull it out. And that's relevant for us too, of course. That's the way we learn different background noise environments. I mentioned the beach. You go to the beach, you learn how that statistics works. Now, we do it as an adult. We don't get exposed to those during our maturational period. But we can improve these things, either this way or through operational conditioning. Okay, let's move on to another plasticity. This is reactive plasticity, which is sort of neither maturational nor associative plasticity. What this is if you have a hearing loss, for example, what happens? Something is changing in your peripheral hearing. Certain frequencies don't occur anymore as they're supposed to be. And as a result, your cortex will change. It will adapt somehow. It will change plastically in some certain ways. So one way is you have a hearing loss, an aging or noise-induced or ototoxicity, whatever, many reasons to get a hearing loss. Or another example is you expose us or the animals to a certain sound environment that is not normal. For example, people who work in a factory or something like this, they're exposed to these overly loud noises all the time. Not only do they get a hearing loss, maybe, but just having exposure to these very, airplane noises, something like this, the brain may react sort of by rearranging something. And here's an example of this. This is a study by Egermont and his colleague in Canada. So this is just, he mapped auditory cortex. These are just the way he did it. And he did it in normal animals, and then he exposed animals, these are cats, to a band of noise from 4 to 20 kilohertz for 3 months. It wasn't very loud, but they were constantly exposed to that. Now, beyond the critical period, this is not critical period exposure. These are adult animals. So this is the distribution of the best frequencies, the characteristic frequencies in a normal animal. Hard to tell, but here it's between 1 and 40 kilohertz. It's reasonably flat. All frequencies, sort of about the same. Well, this is what happens after the exposure. So this blue area is the frequency range of the exposure noise, and now you can see there's a big gap here. This is flat, more or less. You can see overrepresentation below and above, and underrepresentation of frequencies at where this noise is located. So this is, again, just exposure. It's nothing, they don't learn, they're not rewarded, it's not maturational. The cortex will rearrange under these circumstances. We know it's also rearranging, of course, if you're deaf. This is, again, a reactive plasticity to not having any input. So they're going to study that with Professor Reggio, where we looked in cochlear implant animals. And if you have a hearing animal, you deafen them, you put your, acutely, put an electrode in the cochlea, and then you measure what is a frequency preference at different locations in the cortex. You can see this here. Okay, so this is the cortex. This is our electrode that we put in the cochlea, and the different locations here are marked that we measured, stimulating this location or that location, let's say blue and red. If you look at those two cases, blue and red, if you stimulate the blue electrode, you can see lower threshold responses at this location in the cortex. So this is this region here. You see, we would expect a tonotopic organization here. Low frequency, high frequency. So this corresponds to low frequency stimulation. You get something at the low frequency location of the cortex. You stimulate this location, you get something at the high frequency location of the cortex. So normal tonotopic organization in a normal hearing animal, it's still there when you do electrical stimulation. So now you do the same thing in an animal that has been deafened for 5 years. You see a map that looks random. There's no frequency organization anymore. This is the way it would be organized. It would be normal organization. And you see whatever, something that has nothing to do anymore with its frequency organization. And this is, again, there was no reward. The animal was deaf. There was nothing given to it to rearrange, to incentivize its changes. It's a reactive plasticity that is based on some other mechanism than associative operant conditioning and so on. Okay, hearing loss, of course, is one of the bigger issues that we have to deal with. This is a study by Robertson and Irvine very early on, where they made a little lesion in the cochlea at a frequency between 10 and 20 kilohertz. This is the frequency map of the cochlea. But this is now mapping auditory cortex from left to right, and you can see there is sort of a, well, it should look like this going through here in a normal animal, but, well, this was a lesion, so there are no frequencies here, and you can see this reorganization. And in a map, you can see that this region here that is lacking is sort of filled in by some other frequencies that are still there. You see expansion of frequencies. Again, but it's a massive change in the frequency map of the auditory cortex. It depends on the time here. I should very quickly talk about this one. This is really quite interesting because this was a hearing loss, a mild hearing loss on one ear and not on the other. So now input from the two ears goes to both sides of the cortex. They combine on either side. And in a normal animal, the inputs, the best frequency from the left ear or the right ear are matched very nicely. You know, so they're lined up. So what happens when you do look in an animal that has a hearing loss in one ear? Well, these are maps, and this is input from the normal ear. This is frequency low to high, color gradient. This is a blood vessel. We could measure there. It's just one big map from low to high frequency. This is the map now for the abnormal ear, where it had a mild hearing loss, low frequency hearing loss. Looks sort of the same, but you can see here, where it's blue here, now it's orange. The difference between these two maps is here. So these are mismatches between the two inputs from the two sides. Not surprisingly, the frequency inputs are disturbed. This is about 12 weeks after the hearing loss. So now you go to 6 months after the hearing loss. And surprisingly, so this is the hearing loss 12 weeks after. You can see, so one ear input, other ear input. You can see this big cloud. They should be all lined up here. But they aren't because there's this mismatch between the two ears. So when there's 1 kilohertz on this ear, the other one actually has 4 kilohertz. So there's a mismatch. But if you look at 6 months later, that match, this mismatch is largely gone. There are still a few sites. But after half a year, the cortex has realigned those two inputs. Again, this is sort of a reactive plasticity. We don't know exactly why. It has probably something to do with binaural hearing. If you use both ears, you want somehow the inputs to be matched. And so after half a year, it's really quite realigned. And it's even more so when you look at the response thresholds. So this is the difference in the sensitivity. So these are normal thresholds. This is the abnormal thresholds. It's much purple. I mean, there are lots of sites that have elevated threshold because of the hearing loss. That's what you get. So big mismatch between the two ears after 12 weeks. This is a mismatch after 6 months. There's no mismatch anymore. The two ears have adjusted not only their frequency alignment, but also their threshold alignment. The problem is that the good ear adjusts its threshold to the bad ear. That means you have a central hearing loss that is really not in the input. It is in the reorganization of the brain. Okay, never mind. Last one, homeostatic plasticity. This is another type of plasticity which we don't quite understand, but it happens all the time. And that is, you see, so homeostatic regulations has something to do with neural excitability. It's sort of collective phenomena in which neurons are alter their intrinsic or synaptic properties to maintain some kind of target level of activity. It's sort of a bit vague. It's doing something because it wants to avoid too much excitation or too broad inputs. It's something is measured actually locally, and then synapses are being adjusted so that this mismatch or what is wrong is eliminated. So a simple example is this. If you look at structural changes in auditory cortex following a hearing loss, we've talked about these different types of inhibitory interneurons, somatostatin and this and that. And after hearing loss, certain interneurons are downregulated by up to 50%. So you lose a big chunk of your normal circuitry just because you have a hearing loss. We don't know why that is, but that's the case. So now the structural organization is different. And as a consequence, one would expect this homeostatic plasticity to take an effect. So we did a study, actually this measures the different interneuron populations in a normal ear and in an ear with a hearing loss, and from a normal to in the hearing loss, that drop dramatically. This is the same. So, but what we could do is, if you measure the cortex now, you don't know what you see are the differences that you see due to that you have a hearing loss in your cochlea, or is it due that you have this strange imbalance of inhibitory interneurons? So, but we can't separate that out because we have a knockout, a conditional knockout that lowers the number of inhibitory interneurons, just like in a hearing loss, but with a normal cochlea. So what happens? This is work here by Brian Siebold in our lab. So this is missing here. Okay. I can describe it to you. So in a normal tuning curve, you know our V-shaped tuning curve. If you acutely lower inhibition, you block inhibitory transmitter, what happens is this V-shaped thing becomes broader because inhibition sharpens usually the tuning, and it gets to lower thresholds. It's more sensitive. So that's what you would expect with an acute inhibition. So now we reduce inhibition chronically by, we reduce inhibition by some, so we do it now chronically with these, lowering the number of neurons that could provide the inhibition. And what you see is actually the opposite. So this is a normal tuning curve in those animals that have this reduced number of inhibitory interneurons. If anything, it becomes narrower, the thresholds go up instead of getting broader and going down. So this homeostatic plasticity is trying to compensate for the lack of inhibition by doing other things. And what it could do, there are many examples. Oh, here it is, actually. That's the reduction in inhibition in the acute case. Let's skip over this. But what it does is, in this chronic reduction, it probably will just reduces the excitatory inputs. It cannot do much about the inhibitory inputs, but it can reduce excitatory input to keep the balance of neurons, whatever, as it wants it to be. However it measures it, we don't know. Is it number of firing? Is it some other mechanism? But so this homeostatic plasticity takes care of it. Now this is independent from, in a way, from reactive, because reactive changes input. The reactive plasticity is something changes the input. The learning based is changed to the rewarding. This is in adult animals, so it's not maturational plasticity. So it's another type of plasticity. And I will skip this. We're running out of time a bit. So let's talk about a few conclusions. So we have long-term cortical plasticity seems to be governed by 4 different modes, at least. We have maturational plasticity, which is early in life. It's sort of effortless. You know, you don't have to work on it. It just happens. It's automatic. And it depends on the signal statistics and has probably some associative components as well, because these animals are not stupid at their time. They will probably learn some behavioral consequences. But how much of that is balance is unclear at this stage. Then we have our associative learning, a reward-based plasticity that is really in adults. Requires effort. You have to work on it. Getting rewards, being motivated. It's long-lasting, and it's more of a top-down type of thing. That's our reward, you know, telling us we're doing good. Reactive plasticity, following distal differentiation or long-term exposure, has some kind of long time window, have several months, may have a homeostatic component, we don't know. But it seems more of a bottom-up. It's the change in the input that is being altered. Then we have our homeostatic plasticity, following cortical network imbalances, loss of interneurons, long-term window. You know, it's not fast. And it's sort of a local mechanism. So by the way, these mechanisms that implement the synaptic and cellular plastic changes are known as long-term potentiation, spike timing dependent plasticity, and these sort of well-described mechanisms all are part of this. What we talked about is what regulates these mechanisms. They're probably likely shared by any of these mechanisms. Although the control loops that recurrent networks that govern these processes are likely different. As we talked, bottom-up, top-down, local, there are different different governing principles that induce all these changes. So if you open questions, so how can we disentangle the effects of plasticity due to all these different aspects? So all these different modulatory systems. So we really have to start, they're not working in isolation. Whenever we learn something, all of these modulatory systems are involved somehow. We look at one or look at another. It's the combination of them that really tells us how these things work, and we really don't know anything about them, how they combine. So then again, we talked that the inhibitory circuits are critical for this, the different inhibitory interneurons. How do they change with aging, hearing loss, and what is the influence on that, on their behavior on these plasticity mechanisms is still unclear. And in what way do processes that modulate the expression of plasticity, in what way do these processes modulate the expression? As I said, bottom up, top down. There are lots of cases where that we want to restore hearing ability. And we should be able to make use of these plasticity mechanisms in cases where hearing impairment occurs, like in noise-induced hearing loss, aging, of course. Even without a hearing loss, there's now the more hidden hearing loss, early stages of hearing loss that don't show up in your audiogram, but already there, already are changing things in your brain. Not to mention autism, schizophrenia, tinnitus, and so on, which are much more severe changes in our brain. How can we influence those with these plasticity mechanisms? And there are ways, and I think this is the future will open up more venues to be able to manipulate these things. So let's have a brief outlook. It's the last slide here. So controlled induction of plasticity by informed and validated means holds the key to improve hearing ability and more generally brain health throughout life. So plasticity is really key. If we really get a handle on how to influence plasticity, how to manipulate it, so that it really can be utilized to be specific, and that's why it has to be validated and has to be properly applied. I mean, there are lots of these brain training things out there which are not validated. We don't know what they're doing. They claim great things, at least some of them, and one has to be careful with it. But in principle, there is something about those training things, your self-improvement. People like you who go to lectures like this already invoke these mechanisms because you're interested, you're engaging yourself. It's people who are not so engaged and aging just sort of sit there and don't do anything. That's when the negative plasticity takes place. But you want the positive one. You want to keep it up and work on the restoration of your hearing. So good hearing abilities are required for rich communicative function, of course. And rich communicative function is necessary to maintain full cognitive abilities, especially in aging. It's well known that people who have a hearing loss and do not use a hearing aids lose cognitive functions. Their cognitive health declines more rapidly than people who actually exercise or use hearing aids. This is just a simple way, hearing aids. But you can engage yourself in other ways to keep your communicative skills, your brain skills going. You got to work on it. It's all associated plasticity at that level. You got to work on it. It's an effort, but it will keep your mental functions sharper. So as hearing goes, so goes cognition. You know, if your hearing declines, it's a danger that those things will decline as well. Doesn't apply to everybody, but so how do we do this? You know, as I said, improve, maintain hearing, avoid loud environments, use hearing aids if necessary, true, and you can do behavioral listening training. There are lots of things. I mean, learning an instrument, learning a language, all these things will help train the auditory system, will keep you engaged in that sensory system. Of course, there are vision and other things too. And eventually, I think there will be electrical pharmaceutical manipulations that might assist, at least, in that effort. But let's not get too far ahead in that. Anyway, that's the story. And I want to thank all of the collaborators. I think you saw most of their pictures here during the course of the talk. And well, thank you much for your attention.